Welcome, MBChB. Revision Genetics, MBChB. Just tell me hi at the chat. Hello, hello, hello. MBChB, Genetics. Revision, MBChB. Just say hi as you pass. Today we are meeting with MBChB. Genetics revision in preparation for our cut. Hi. Rooney Advik. Hi. I know I've come a bit earlier, 5 minutes earlier than the normal time, so that we can finish early. So we have to start early. MBChB, hi. MBChB Genetics, thank you. MBChB Genetics Revision. Just say hi. Just say hi if you're there. MBChB, hello Salsa. Hello, yeah, just say hi, MBChB Genetics Revision. Today we are meeting with MBChB online, Genetics Revision Genetics, here too. Welcome, just say hi. I can see AYSA. I don't know how to pronounce it, but hello, Salsa, hello. Elvira Gomez, hi. MBChB, just log in. I came a bit early today because we have a lot to cover and possibly we won't finish today. So we have to start early and we have to agree whether I start with Hardy-Weinberg or I start with Mendelian. We are going to vote and we know how you want me to start. Should I start with Hardy-Weinberg? Hello, madam. Thank you so much. I was your student for biochem and I passed. Sheila Paranai. Yeah, I can remember the name. BMLS upgrading, right? Thank you so much. Much appreciated. I'm humbled, my dear. I'm happy that you passed. You can still just stay on board if you feel like listening to what Madame is teaching. You are most welcome. But I'm happy that you passed, and I can see you are one of my best fans. Huh? So it shows the crown. I appreciate. So this far, it's good. We thank you so much for everything. All my students, for the support you've given to me, we'll be able to make it work. So we've already been able to simplify biochemistry. So I'm happy about it, Sheila. I can remember the name. I think it was BMLS upgrading. Though I can't remember the intake, but I appreciate so much. You are welcome. MBChB, can we log in? I'll only give 5 minutes, then I'll start. You'll join as we proceed, because we are not going to wait the whole day. What pattern? Pattern. MBChB, can you log in, please? We start. You agreed that it was today, right? Because the class rep told me you agreed it's today. And you can't change it. If you change it, I won't be available. Because I'm also sacrificing my time so that we can revise. Yes, but we'll start at that time. I agree with you, Salsa. I agree with you. We'll start at exactly 9:05. So whoever want to join will join, but I want the opinion of the group. Because we have a lot to cover. Whatever you saw I sent, I separated them into subtopics which we've been covering. The way we've been covering them, the chromosomes, the genes, Mendelian genetics, a bit wide. Then we have the Hardy-Weinberg. So, Salsa, together with the others, can you tell me if we can start with Hardy-Weinberg? I'm fine with either way, or we just start the way it shows, because it seems we might not finish. If we go today, we can go even for 3 hours, which I can, but I don't know the side of the students, if they'll be exhausted. I can discuss everything, the 5 pages which I sent, but I'm waiting for the opinion of the students, but they are coming very slow. So I'll start. Salsa, what do you say? Let's start the way it shows, kindly. Very good. That's what I want. You know, nowadays people have opinion. You don't just decide things and you say, okay, there are cases where you can decide and it's final. But there are cases where you have to involve the opinion of the other parties. So I can see Wizard has just answered me. I'm already starting. We are at 20, but I'll start because maybe other people, they are not interested. Maybe they already know. So for somebody who feel they are already fine, they can face the cat, no problem. But there are people who are willing to know more so that they can know how to answer questions, which is fine. I told you it's not Madam Winnie to want the likes. No. All of us, we have to put sacrifice. We have to sacrifice our time to pass. When I can see the students understand what I've been teaching and they put it into writing in the cards and the main exam and they perform well, that is all I need. That is how happy I'll be because I shall have already done my part. So, Wizard, we can start. Jenny, you are there? Tell those students that we are starting. Before you could come, I said that we were to decide whether to start with the Hardy-Weinberg or we do the way it's showing. Wizard has just said that we start the way it shows. In the part of introduction to genetics, where we have chromosomes and genes, I'm not going to take much part on that because it is self-explanatory. But on that note, I'll say, question 1 says, I think I skipped. The question 1 is where it reads introduction to genetics, which is fine. But the question 1 is the question 2, which is there. It says classification. Discuss classification of chromosomes based on the position of the centromere. You know, that one is simple. You have to define the submetacentric, the metacentric, the acrocentric, and the telocentric. So I said you have to stress much on the position of the centromere, and then you explain much where it is, the position of that centromere on the chromosome, and then during anaphase, which shape does it show? So for each of them, each of the 4, and then you draw. So that's how we are going to break down those marks. And it has to go to a maximum of 10 marks because I'll be giving half-half. The same with the genes. 7 classes of the genes, we did them. You have the pleiotropic, you have the lethal, you have the basic genes, you have the multiple genes. Just define, put the name of the gene or the class of the gene and then define. That is simple. So that makes me to go straight to the monohybrid genetics. Mendelian genetics, I start with the monohybrid cross, the way they're showing. So from question number 2 to number 5, to number 4, sorry, nothing to talk much about because it is self-explanatory. You'll be able to explain those chromosomes based on the position of the centromere. The genes, you already know, That is something easy. So let's dive straight into Mendelian genetics. Question number 6 says, in border collies, black coats, which is dominant, capital B, it is dominant to red coat, small b. We said we are talking about biallelic. Didn't we see you today? Where? Jenny, today I was not around anywhere. I went to church. In Thika. Or the question, I don't understand the question which you are asking, but it's okay. So, we are talking much about the border collie, where we are talking about the color, or the coat color as the trait. So, we have the capital B. I told you, write for me clearly, traits, coat color, alleles. Write clearly, whatever you can see. Come here. Give me capital B, black. If you like, you can repeat coat color. I won't mind. Red. Let us draw the Punnett square first, then I can go back to the question. I told you you have to include for me the male and female. Don't give me a Punnett square without the male and female. So we are saying, Charity Wekesa, welcome. We are saying that in border collies, the black coat B is dominant to red coat, small b. A breeder has a black male, which has done what? It has won numerous awards. The breeder would like to use the dog for breeding if he is purebred. If it is purebred, it means this dog was coming from the parents' pure breeding. Is among the heterozygous, the hybrids which are produced here. What does the story say? The story says that the breeder would like to use the dog for breeding if he is pure breed of BB. To learn this information, she test crosses. The breeder is a female. She test crosses him. Him is the option there where you can see the hybrids in the F1s. She test crosses him with a red female. One of these is that boy who has won an award, that dog. It means it is test crossed by who? By a red one, homozygous recessive. This is the male who has won an award, who is being test crossed. So you will have then let's look at the question. These are the F1s. These are the F2s. The question says what? It says, after test crossing with a red female, so you need to have the two Punnett squares, the one where it has the parental generations, then the second one where you do the test cross. So you are saying, the question says, draw a Punnett square for F1 and F2, which is already on the board, and state the genotype and phenotype of F1 and F2. The genotype for F1, what we said, all alike. In bracket, write capital B, small b. We have the genotypes is either homozygous dominant, heterozygous, or homozygous recessive. So don't write it in words. Write it the way it shows there. All alike. Phenotype, all black. All alike in bracket, black. F2, genotypic, or the genotypes are, you see, wherever we were doing this, you had a comma. And now you are talking about the phenotype black, comma, red. If you are asked about ratios, put the colon. If you're not asked about ratios, don't put a colon. Nobody needs a colon. So you are asked about the genotypes in F1. They are all alike, bracket capital B small b. You are asked about the genotypes of F2, 2 capital B small b, comma, 2 small b small b. Or you can say capital B, small b, the heterozygous is represented there, and homozygous recessive. That brings the phenotype of F2, black, comma, red. Simple. Answer what you've been asked. Question number 7. Question number 7. A pea plant. A pea plant is the F1 offspring. A pea plant is the F1 offspring of true breeding plant with purple flowers and a true breeding plant with white flowers. Trait, flower color. Trait there at number 7, flower color. Describe something which makes sense. Don't just write color. Of what? Flower color. Flower color. Alleles. Capital P for purple. Small p for white. Let's hear the story. Draw a Punnett square. Then I can proceed with the story. We agreed the male up. Female will be this side. Let's see what happens. They are saying a pea plant is the offspring of true breeding plant, purple flowers and white flowers. Small p, small p. It means you are going to have the hybrid. Then, this plant, which plant? The offspring there, is crossed with one that has white flowers, recessive. It means it's still a test cross. One of those F1s, the hybrids, will be test crossed. One of them will be test crossed with homozygous recessive. Then you are asked to draw a Punnett square. Of course, you have to draw for me a Punnett square. Don't skip by giving me an answer without a Punnett square, because the Punnett square will always... Genotype in F1, we can't write 4BB and leave it at that point. That is also fine. Jenny, the 4BB is still okay. All alike, you write in bracket capital B small b is also fine. But you'll have to mention the 4 because there are 4 there. Even if I talk about all alike, all of them, there are 4 in those boxes. So that is okay. You are still correct. But now, if you write for me capital B small b without mentioning the 4, without mentioning all alike in bracket capital B small b, that is wrong because you've left an incomplete statement or an incomplete answer. So you can write 4 capital B small b, you will get mark, or all alike in bracket capital B small b. The word all talks about all the 4. So either way. So we are talking about question number 7, and now we are trying to answer. Pure breed of the pea plant, purple being dominant, white being recessive. Pure breed, homozygous recessive, homozygous dominant, brought the hybrid. From the hybrid, a test cross is done. That is what we have here. So you have to have the 2 Punnett squares. So you have to draw the Punnett square. There is no shortcut, because from the Punnett square, that is how I'm going to tell whether you know how you came about to the answer. And it starts from up. If everything is wrong up there, especially for the diseases, then everything will be wrong down here. If you place for me an allele for the dominant recessive, for the autosomal dominant instead of autosomal recessive, up there I'll know everything will be wrong. So you have to present the trait clearly there, the alleles. That will be the what will be guiding me to know whether the Punnett square is correct, whether the answer which you are giving me, you know, you have not copied from a neighbor because you saw a neighbor writing that answer. It is your work from up there. You understood the question well and answered it correctly. So the question asks how many of the plants in the next generation will have white flowers? How many? 2. We'll have 2, purple 2, white. So the white flowers will be 2. If you talk about 2 out of 10, out of 4, half of them will be white and half will be purple, because the law of dominance, the purple one will suppress the recessive allele and those 2 will show that they are color purple. Question number 8. Ability to curl your tongue up, which is known as tongue rolling. I'm going straight to write the trait so that we can understand what the question says. Tongue rolling. Write for me clearly. The trait, tongue rolling. Can roll. Something like that. Cannot roll. Cannot. Don't write tongue rolling present, tongue rolling absent. What is tongue rolling present, tongue rolling absent? Can roll. Cannot roll. Simple as that. Simple English. We are saying, the question says, the ability to curl your tongue up on the sides is dominant to not being able to roll the tongue. Now the question comes, a woman can roll her tongue. A woman who can roll her tongue. This woman is either heterozygous or homozygous dominant. Let's see. So let me draw the Punnett square first. We understand more of the story. The males were starting up here, so we still don't know much about the male. Let me see. Or is a female which we still don't know much about. Let me see. A woman who can roll her tongue. Man is a man who cannot. In fact, the man is the one who has the story. Well, the man has to be homozygous recessive. The man cannot roll the tongue. A woman who can roll her tongue marries a man who cannot. Their first child has his father's phenotype. There is a child in the F1 who will be small t, homozygous recessive. So we don't have any other option but to put the woman as heterozygous. Then you will have so you can see 2 of the children cannot roll their tongue, 2 can roll. So they are saying that child whom they are saying that has the same genotype as the father cannot roll. Brings us straight to put the mother as heterozygous. Because if you could have put the mother homozygous dominant, then all the children will be able to roll their tongue. So the mother is heterozygous, the father is homozygous recessive, and the child is homozygous recessive. Simple as that. The Punnett square is going to help us solve all the problems. That is what it said. Question number 9. How does this question go? Create a Punnett square for a cross between homozygous purple flowering plant and homozygous white flowering plant. What is the phenotypic and the genotypic ratio? I think this one, homozygous dominant, correct. So this one relates with question number 7, but question number 7, it was a test cross of what? Homozygous recessive. So in this case, it is homozygous dominant. We talked about flower color. They will say, Madam did not write that one. I'm writing. So it's the same as the one which we had. Purple, white. Still, you will have the first Punnett square where we generate the hybrids because they talked about pure breeds. But then, of the hybrids now, you do a test cross using homozygous dominant where you will get this, this, this. All of them will appear purple. But now, the genotypes, you will have 2 capital P, 2 capital P, small p. Phenotypes will have all purple. Or only purple you'll have. So for all the 4, it will be purple. So we are talking about question number 9. That's what I did. It relates with the question number 7, but now you are doing a test cross, or it talks about what is the phenotypic ratio and the genotypic ratio. The genotypic ratio, if they talk about the genotypic ratios, there has to be a colon in between here. 2 homozygous dominant, 2 heterozygous. Then you talk about phenotypic ratio, all alike, in bracket, purple. Question number 10. A monohybrid course on parental strains based on seed shape, round capital R, wrinkle small r as a trait generated as F1, which were all carriers of the trait. This is just the same. You have pure breeds of round, cross with the wrinkles. You will get hybrids, heterozygous, which will show they are all round. Then do a test cross using homozygous recessive, whatever we did in the previous one. You will have the genotypes and the phenotypes as it's spread out, the way we had the purple one. So it's just the same applicability as the one which we had for question number 7. It's only now the trait is the seed shape. So you just apply the same. Now let's dive into Mendelian disorders to understand much on how the Punnett square works. And we are going to start with the X-linked. And if you look at those, the first 3, are they the first 3 or the first 4? The first 4 are X-linked recessive from number 11 to number 14. Number 15 is X-linked dominant. So let's start with the first one for the diseases, because I like testing on diseases more than the normal monohybrid. You know, monohybrid can be very easy. But for the diseases, Mendelian diseases, it is up to the student to know whether now Madam's question relates to X-linked, autosomal dominant, autosomal recessive. That's how it goes. Now, the question says, in a family, father is hemophilic. Let's start. Write the disease here clearly. We'll know. You are working on hemophilia. Hemophilia. Write for me hemophilia. Then I said, alleles. Since it is X-linked, put for me the X. Why are you leaving the X out? When it has X and small h, say present, because the allele for the disease is recessive, but the disease affects the sex chromosomes. It means the X capital absent. Let us put the Punnett square and follow the story, where the story is going. So the male is here, up here the female is here. Let's read again. Father is hemophilic. It means the dad, Y is not touched. Please don't put anything on Y. Father is hemophilic. Small h, sorry. And then the mother is what? Is a carrier of hemophilia, a carrier. The mother is a carrier. Look at the options. Look at the options. What does the question say? You can see we have affected girl, affected boy, healthy girl, healthy boy. As much as that girl is a carrier and the allele for the hemophilia is recessive, just as we have the autosomal recessive disorders, they show they are unaffected because the allele for the disease is recessive. So for that girl, it is not affected, or she is not affected. The affected one is down here. 2 of the children are affected. 2 of them are healthy. A male and a female affected. A male and a female healthy. So the question says what? State how many sons and daughters are affected, not infected, and healthy. 1 daughter, healthy. 1 boy, healthy. 1 girl, affected. 1 boy, affected. 2 children are affected. 2 of them are healthy. The allele for the disease is recessive. The allele for healthiness will suppress the allele for the disease. So in heterozygous state, for the X-linked recessive, they will show they are not affected. Question number 12 relates still with hemophilia. But now, we are going to leave the trait the way it is. We are going to leave the alleles the way they are. Don't interfere. Let's go to our Punnett square. We see what we can change. A hemophilic family involves a man who is healthy. Let's change the positions. The man now is capital H. And a female who is what? A wife is still a carrier. So it means a girl will come healthy. A boy will come healthy. There will be a carrier girl who is not affected and 1 boy affected. Then the question says, draw a Punnett square, show the F1s, of course. State the gender and number of kids who are healthy. The gender who is healthy and the number of kids. Males and females are healthy. 3 kids are healthy there. Only 1 is hemophilic. Which gender is hemophilic? Male. As you can see there. The Punnett square will solve all our genetic problems. Question number 13, DMD. It is still under X-linked recessive. In humans, Duchenne muscular dystrophy, DMD, is a disease where the muscles become weaker over time. Full stop. The gene is located on the X chromosome and is recessive. You know, when I'm setting, I'm not going to explain to you all those. I'm going to remove that part of recessive because that one is spoon feeding the students too much until they know it falls under the X-linked. I'm just going to stop at DMD. Then I follow the one which says those afflicted with this disease usually die in childhood. If a female is a carrier for DMD, married to a healthy man, it means, let us change our H to small d. Let us change here to DMD in short. But that is your day, right, for me in full. Capital D, disease absent. Now, the man is healthy. I think that is what the statement says. Let me see again. Healthy male, a female is a carrier. Just change H with a D. Then we will have a girl who come out healthy, a boy who come out healthy, a carrier girl, and affected male. It means 3 of those kids are healthy. They are not affected. 1 boy, 1 child is affected. So, what percentage, good, they talk about probability, of her sons will inherit the disease? What percentage? 25%, because only the affected will be able to transfer the disease to the next generation. What percentage of our daughters will inherit the disease? 0. We still have to stick to the law of Mendel. These are Mendelian disorders. So the law of dominance still have to apply. The way it applied in hemophilia, it's going to apply in DMD because they fall under the autosomal sex-linked recessive disorders. Sorry. What percentage of our daughters will inherit? 0. Percentage of the boys which will inherit? 25%. In other words, they talk about the probability that the girl will inherit the disease. Probability that the boy will inherit the disease. So it means that it's also the same as percentage. Colorblind also falls under the same class of disease. So for the colorblind, we talk about if a man is colorblind. Give me up here, colorblind. Don't say, madam, you are the one who mixed it up like that. Colorblind. Let's proceed and see. So also small c, capital C, present, absent. If a man is colorblind, what percentage of his sons will be colorblind? Assuming the mother is not a carrier. The mother is not a carrier. It means the mother is healthy. Capital C, capital C. But the father is what? Colorblind. We'll have, this child is not colorblind. This boy is not colorblind. This child is not colorblind. But there is, all of them are fine. Yes. That's how it is. Because the mother is showing that he's healthy. She's healthy. Only the father is showing that he's colorblind. If the man, what percentage of the sons will be colorblind? 0. 0%. You can see. They don't have any allele for the colorblind. The last one, number 15. Number 15 relates with sex-linked dominant. And if you saw the examples which we give during the lectures, it was the only one which I gave there, the vitamin D resistant, the rickets. So if a man has vitamin D rickets resistance, what percentage of his sons will be affected, assuming the mother is a carrier? And I'll always tell you to use R, use D, use T, use H, for uniformity. So that if you come with your own letter, I'm not going to mark. I'll have to tell you use this letter. So that you don't come with your own, please. So, Wizard is putting question mark. I'm also reading the charts as I go. I don't know what the question mark is all about. So the trait is, you can give the rickets the way you want it, but now it's going to change. Here, we are going to talk about capital R, where you say present, small r absent. And they are saying that assuming the mother is a carrier, capital R, small r, the father is what? It has the disease, so capital R here. And in this case, if it is a carrier for the offsprings, it means that one is affected. That is how the law says. Capital R, small r. Only the boy do not have the disease. One boy, which you can see there. One boy do not have the disease. The other 3 children are affected. So the question says what? What percentage of his sons will be affected? The percentage of the sons who are affected there is what? 25%. Is it of the children or of the sons? Percentage. 1 out of what? 1 out of 2 or 1 out of 4? If we talk about the children, then it's 3 out of 4. Only 1 is not affected. So if you talk about the children who are affected or the gender which is affected, 2 of the sons, 1 will show affected, the other one is not affected. But of the children, 3 out of 4, 75% of the children are affected. Only 25 is non-affected. Autosomal dominant. I separated them very well, so that you can also follow when you are revising. You can clearly remember which class the disease falls under, so that you can guide me straight from their alleles. Yes, 50%. So we are talking about autosomal dominant. You see? Perfect. Whatever was written here on the board, or whatever I've written here on the board, relates with sex-linked dominant. We are now talking about the autosomes are the ones which are affected. But then the disease allele is dominant. Whatever is on the board, the sex chromosomes were the ones which were touched. And the allele for the disease was still dominant. So it means the law of dominance is still going to apply. But the difference with autosomal dominant, please don't put for me the sex chromosomes. I'm going to rub everything here so that we don't confuse it. So I'll use the example for the autosomal dominant, the Punnett square for all the autosomal dominant, just as I use for the sex linked for all. So we are starting with the first one. It says achondroplasia. Give me the trait here. Achondroplasia. And then the alleles, please. Don't put for me the sex chromosomes. Come here, capital A, present. Small a, absent. Then, let's get the Punnett square. We hear the story. What happens? male, female. Let's see. They are saying heterozygous male for achondroplasia mates with unaffected female. Unaffected female. It means it's homozygous recessive. Heterozygous. Was it heterozygous? Male. Let's see. Half of the kids are affected, half unaffected. So it means the question says, list the number of affected and unaffected children. I told you for the autosomal dominant and autosomal recessive disorders, I don't focus on gender because the disease affects the autosomes. So I just talk about the affected children and the unaffected children, or the ratio of affected children to the unaffected, or the probability of affected and the probability of unaffected. The children. I don't talk about the gender. So you can see clearly there, Punnett square will give you everything. In fact, from the Punnett square, if you get it right, you can get all the genetics problems getting solved. You can get them, all of them correct. So heterozygous male met with unaffected female, using A for list. A for, what is that for? Using A, comma, list the number of affected and unaffected children. So it's 2 children affected, 2 unaffected. Number 17, still autosomal dominant. So I can set any of those diseases, which I don't have in the examples, only for you to apply the same. It can be osteogenesis imperfecta. It can be neurofibromatosis, any of those. As long as it falls under the autosomal dominant, I can use any of the examples. But as long as you understand the genotypes of the parental generations, what it will come out from, the cross between the parental generations and the F1s, you'll be able to answer the question, any of the question which is given. So still heterozygous male, but now we are talking about progeria. Progeria. I told you this is also a disease which shows the child appear to look old. So capital P, sorry, I rubbed everything, but capital P present, small p absent. Then the story says what? Heterozygous male for progeria. Unaffected female. It looks the same as whatever we had. Unaffected female. So you will have the same as whatever we've had in the previous one for achondroplasia. 2 of the affected, 2 non-affected. Because the allele for the disease is dominant, the heterozygous will show affected, and the homozygous recessive, they only get expressed when you have both alleles, the recessive alleles. So they only get expressed in homozygous recessive state. So that one you have 2 unaffected, 2 affected. Autosomal recessive. Question number 18. In autosomal recessive, what we discussed in class, they are not expressed in heterozygous state, just as what we can see in autosomal dominant. They are only expressed when they are homozygous recessive. So that is what we learned. So let's start with the example. They are talking much about sickle cell anemia. So you give sickle cell up here, talk about sickle cell. Don't put the X and Y's, please. These are for autosome. Capital S, absent. Small s, present. Punnett square, male, female. You have to include the male and the female. If you don't include, I'm not going to give you a mark. I'm going to cancel the whole Punnett square. I won't even be lenient by saying that, because I don't know what you're giving me there. If you don't include male and female and you expect to get marks, and even if you interchange the alleles there, everything will be wrong, plus your marks, plus the answers. So it has to start from up there. You understand the question well and answer it correctly. So we are saying, a woman heterozygous for sickle cell anemia with heterozygous male, heterozygous female, heterozygous male. We will have unaffected, 75%. Affected is 25%. And that is one of the characteristics as well. The affected, they are always, majority is 25%. So list the number of affected and unaffected children. 3 unaffected, 1 affected. You've been told the number. List the number. Just say 3 unaffected, 1 affected. Simple. Then you go to the next question. Cystic fibrosis, the same. It is caused by a recessive allele on chromosome 7. Now you know much. The group which was working on cystic fibrosis, you know much of the symptoms, treatment, how it come about. But then it is caused by a recessive allele on chromosome 7. A person must inherit 2 of them. It means they are only expressed in homozygous recessive state. So it's caused by a recessive allele on chromosome 7. A person must inherit 2 of the alleles to have the disease. What are the chances of a child having the disease if both of the parents are heterozygous? You can see now, it's the same there. You'll write cystic fibrosis. The alleles will be the same, capital C, absent, small c, present. If you have the parents heterozygous, the chances of getting affected child will be 25%. So that's a bit, it relates with whatever we've been writing, of having a child if both of the parents having a child having the disease. So the affected will be 25%. The other one talks about Mendelian single gene disorders. So if you could remember well, I was trying to arrange the questions in relation, in order of our lectures, so that we can easily revise or remember. So after that, we looked at the chromosomal disorders, which were caused much by mutations at the level of the DNA. And then we had the multiallelic, which I didn't include here. So you can see the examples of the diseases which we worked on. Or question number 20 talk about the 2 Mendelian disorders. I think in this case it's too wide, because if you talk about the 2 Mendelian disorders, what I need here is you can either take the autosomal dominance, you talk about the characteristics, and you give example, a case of 1, what did I say, 2 diseases, 3 examples of the disease in each case. If you take autosomal dominant, give example of 3 diseases just by example, and then you can do a Punnett square, assuming both parents are heterozygous, or in a scenario where you have the parental generations, the allele for the disease is dominant, and the other one for the healthiness is recessive. So using the Punnett square, you can give example, mention 3 of the diseases, but you can give example in a Punnett square of the same. Then you can also do the same for the autosomal recessive or the X-linked. So you can do autosomal recessive with X-linked recessive, because that's what we discussed much. The X-linked dominant is not much. Or you can do autosomal dominant with X-linked recessive, but you name any of the 3 diseases, because we have much. In that table which I gave, there are several diseases. Then you give the examples based on the Punnett square in a scenario where the parental genotypes of the parents could be heterozygous, could be homozygous dominant, and then one of the parent could be heterozygous. Then what is the outcome of the F1? Such scenarios. That is the question 20, because it talks about 2 Mendelian disorders. Question number 21 is what I was talking about before I could see question number 20. The chromosomal disorders. Remember, we talked about the Klinefelters, where they look more like males. They are biologically males, but there are some problems because there is an extra X. So we saw such characteristics. And then there was Turner syndrome, which relates, they come out as biological females, but we saw some of the characteristics which was relating to such disorders. Then we had also several, the Criducats, the other ones which we could also see. So you can discuss any of those if you are asked about 4 chromosomal disorders. So that is something which somebody can read and understand. The ones which can be read and understand, I don't take much time discussing them because with them you can understand. But the ones which need a little bit of concentration and understanding by drawing the Punnett square and try to see how we can tackle the question, those are the ones which I can take a bit of the time to discuss on the board. So it still talks about writing short notes on the Turner syndrome, writing short notes on the Klinefelters, but these were falling under the chromosomal disorders. So I'll go straight to the dihybrid cross. And as I mentioned earlier, the main point for dihybrid cross is we are trying to look at the law of independent assortment. At the point of segregation, you have 2 genes which control 2 different traits. The gene which controls one trait do not depend on the other gene which controls the other trait. So we are talking about independent assortment. And at that point, you have those 2 genes, then definitely you need to have the 4 alleles. So after identifying the genes, you are going to have the alleles. You have to generate the alleles. At that point, when you're generating the alleles, you have to have the gamete combinations. And when we are talking about the gamete combinations, when you talk about the gamete combinations, remember we were using the heterozygous. The heterozygous, the number of heterozygous, you'll talk about the 2 power n. N is the number of heterozygous in that case. So when we have 2 genes, it means the first gene will have 2 alleles, contrasting characteristics. And then the second one, you'll also have 2 alleles. So we are talking about 2 power n. It means that we are going to generate 4 of the gametes. So the main point here is to exercise the independent assortment law. So when I have the first example, then it is going to apply to all the 3. It's going to apply to all the 3. Then we can do Hardy-Weinberg. I'll do the number 1 of the dihybrid cross. It says gene A controls fin length. And if I talk about gene A and gene B, kindly don't come with your own letters. You'll start telling me G, capital G and small g. Just use the A which is there. So we are talking about trait. We have fin length, trait 1. Fin length. Fin length. In fin length, we are talking about gene A. Give capital A, small a. So for the capital A, we are talking about short fins. Short fins are dominant to long fins, meaning short, long. Trait 2. we are talking about fin color, or gene 2 or trait 2, whichever. Fin color, it is B. We are talking about gene B. Let's see. It says gray is dominant over lemon. Give gray, give lemon. We said the next step is we are going to generate what? The gametes. In all those cases, even if they are pure breeds. But then you are going to have hybrids. At the point you are crossing these two, you cross these two. You are going to have the hybrids as the outcome. So it means those are two different alleles. Then you have this with this. This one will go with this. You'll have AB. This one will go with this, AB. This will go with this, AB. This will go with this. Sorry, this will go with this. Those are the gamete combinations you'll have. Who is this? Please don't comment things which are not here. MKU Med 2024. Can you just get out if you don't want to concentrate? People are concentrating, kindly. I don't know where the hot water is. Who hot water? This man's fever is no joke. Oh, you had the one who was coughing. Ah, yes. Thank you. I was wondering whether you are talking to a patient and we are in class. Thank you for that. We are going to give it. Yeah, I'm sorry. I'm sorry for talking bad. Yet you had the child coughing. So it's fine. Thank you for that. I'm going to give. So if you talk about the gamete combinations, then you can see cross capital A with capital B, capital A with small b, small a, capital B, small a, small b. The gamete combinations, we have 4. Because the alleles are how many? They are 4. So you talk about 2n, because you can see up there we have the first heterozygous, second heterozygous. So 2n will bring to 2 times 2, where we are going to have 4 gamete combinations. So with that, let us draw a Punnett square. That is what we said. So after drawing the Punnett square, you will apply to all. I'm just going to rub because, you know, the Punnett square will carry a big space. Give me the male here. We agreed the males have to be here. The females have to be here. But make the Punnett square. Because the final question you will be asked is, give us the phenotypic ratios or the phenotypes of the F1s. So we have the same for male and female. For male and female. Now we can fill it. We can fill the Punnett square, and I told you don't gaze. If you gaze, I'll know. If one of them is wrong, then everything will be wrong. Because there is no way you'll tell me 9331 if one box is not done correctly. We are at the 3rd row. We are at the 3rd row. Then we are going to see So we need 9. We need 9 gray, short gray. We need 9 short gray. We need 9 short gray. We need 3 short lemon. We need 3 gray, what, long. Then 1 gray, short. No. What do we need? What is A, B, B? A is long. We need 1 long gray. Correct. So let's see. 9 short gray. We'll give a tick. 1, 2, 3, 4, 5, 6, 7, 8, 9. We have 9 short gray. Then short lemon, it means capital A, small b. Capital A, small b. 1, 2, 3. Then we need long gray. Small a, capital, long gray. Did I get it right? Long lemon was not there. So long lemon is the one which is 1. So long gray. Long gray, we have small a and capital B, this one. So they are 3. And then lastly, we have long lemon. 1 long lemon. Yes, because for length, short was dominant. Long was, can I write the alleles again? This was long. This was short. Capital B was for gray. Small b was for lemon. So we talk about, capital A was short, small was long. Long lemon, 1. Jenny, is that one correct? Long lemon is the one, right? I need a response from your end, Jenny. We got them right because short was dominant over long. You know, we always know that the long one is the dominant one. But in this case, you could see short fins are dominant. So the long fins are not helpful to the, yes, long lemon. Thank you, Esther. So it applies to the other ones, number 2 to number 3. Because what we are trying to look here is, are we able to see or to exercise on the law of independent assortment, where 2 genes control a trait, but they are independent of one another? So just as we agreed in class. Yeah, thank you, Charlene. Agreed. Vinke, thank you. Oh yeah, I had left it out, correct. Now I had to put the alleles. And that's why I'm telling you, if you put the alleles well, it can easily guide you, even you yourself, from the end when you are writing the answers as a student, and even from the side, which I'm marking, because I'll be able to know that you know what you're talking about. So you will be led by the alleles, the traits, or the genes which control such traits. And then definitely you'll clearly generate the gamete combinations, and then you put them in a Punnett square. Then you'll be able to answer the questions. They're talking about the phenotypic ratios. The phenotypic ratios are the short gray 9 to 3, 9, 3, 3, 1. So you talk about 9 short gray, colon 3 short lemon, colon 3 gray long, colon 1 long lemon. So they also asked about the phenotype. You know, the phenotype is different from the phenotypic ratios. You'll tell me the phenotypes are short gray, short lemon, gray long, long lemon. Those are the phenotypes. But if I ask about the ratios, the phenotypic ratios, the phenotypes are short gray, but now the ratios are the numbers. 9, 3, 3, 1, but mention the phenotypes. So it applies to the other, number 2, number 3. What I want to see is, do you understand how the law works? That's how simple it is. So it's good we can do the Hardy-Weinberg, because then I can leave you people to go and revise. I don't have any problem. But we need to concentrate, because I'm already starting on Hardy-Weinberg. So we just did it the other day. And I told you I'm going to focus much on the serious parts, where the students can make a mistake. We stress much what the student need to write, or what the student need to think of when they will have to write the answer. We are starting by calculation of heterozygous. Remember, this is where we talked about genetic counseling. For most of these diseases, which are autosomal recessive disorders, we want to see in the next generation how, whether the allele for the disease is going to be represented the next generation, or it will be going to extinct. But remember, as much as we are looking at the heterozygous, because for the homozygous recessive, we know clearly that these patients are affected. You'll clearly know, or you directly know, that those patients are affected. But in the case where we have the heterozygous, there are traces. So there is no given point. One of the traits can have a homozygous gene in dihybrid crosses, homologous gene. What do you mean by homologous gene? Because they're talking about those genes being independent. As much as they represent the traits, they express the traits which we are looking at. They don't depend on one another. Vinke, the question, I'm not understanding it well. I'm not understanding it well. Yeah. But you'll be able to explain to me, possibly I'll be able to understand. But we are talking about the traces of the disease allele lies in the heterozygous state, because we have both the dominant and recessive. So let's start by calculation of heterozygous. That part which has a problem is what we want. We don't want the part of the people who are fine. So they're talking about testing PTC. It's due to a single gene called T, the dominant T. We don't want to calculate anything on the P. The P people are fine. We need the Q allele, this few percentage of the people who can't do things right. Those are the ones we are intended to know much about. So they're the ones which we are looking for in this heterozygous state. So they're talking about they can test PTC, which is fine. You sample, the number of N is 215 individuals. 138 can detect the beta PTC. We don't have a problem with the 138. Why would a student still tell me 138 over 215 and then to get the square root? I don't need that one. I need the people who cannot do it correctly. So we are seeing that 77 could not. I'm interested in 77. I'm not interested in the other ones who could test, because with them they are fine. What of these people who cannot identify a color? These people who cannot test? These people who have failed from a test? Those are the ones I want. I don't want the people who passed. I don't want the people who can test. I don't want the people who can clearly see the color. The ones who cannot see. And you have to see that the total, it should add to total. They're talking about 77. If we add to 138, it brings to 215. So we are talking about the PTC. In this case, we don't talk much about the Punnett square. We are calculating. So you do 77 over 215. And we said those are the minority. They are Q squared. I had to carry my calculator with me, my tablet to calculate, because I'm projecting using my phone. So it is good if I calculate from my end, because if I wait for the students to calculate from their end, then we can stay here forever. And where is this calculator I'm bragging about? Do I even see it here? So we are going to do 715 divided by 715 divided by, just a minute. Where did the calculator go? I don't even know. 715 divided by 77 divided by 215. Oh, perfect. Please, if you give me answer from that end, I'll be very happy. Good. 0.035. Can we stop? If we stop, yes, 58. Is it 58? 0.03558. Let's do that. 0.03. Kindly, you'll be able to give me the answer. I'll read. No, wait. Somebody is saying 0.358. Which one is which? 0.3 or 0.05? I can see majority talking about 0.358. Agreed, please? Now, can we find the square root? Kindly find the square root, or you already found the square root? Perfect. 0.358. Can we find the square root? That is the square root. Thank you, everyone. Can we find the square root? Is it the square root? First, I want us to divide 77 over 215. I hope it's a 0.3581. Yeah. Can you do the square root now? Oh, good. Thank you so much. 0.598. Thank you. Our Q is 0.598. Then the rule was saying P plus Q equals to 1, meaning we are going to look for the other allele, the dominant allele. So you say that P plus 0.598 equals to 1, meaning P equals 1 minus 0.598, P equals to 0.4 what? We can subtract. Thank you. 0.402. So we have the alleles. First and foremost, that part which you want to know, the one where we have a problem. Thank you so much, everyone. Divide it with the total number of people in that population. Find the square root, because that is Q squared. That is homozygous recessive. But then at what point do we need the allele? For Q, do the square root, we are getting 0.598. But the allele frequencies, they should total to 1. So P plus Q should be equal to 1. Just subtract 1, 0.598 from 1, and then you'll be able to get P is 0.402. Let's see the question. Calculate the allele frequencies of each allele. Already, if you clearly do this, I'll be able to see. 0.598, the recessive allele. Dominant allele, 0.402. Then the question says, leave alone the expected genotypic frequencies. That one cannot be 4 marks, because what you are going to do here is, from the Q which you found, Q squared times the total number of population, P squared times the total number of population, whatever you are doing, you could remember. 2PQ times the total number of population. So that question, which you see the expected genotypic frequencies, I can ask it, yes, but it's not for 4 marks. 4 marks reaches where we have reached. If I have to add another 4 marks, then I can express the expected genotypic frequencies. The question is just correct, but the marks is less. So if you are asked about the genotypic frequencies, for example, the only genotypes we know are the Q squared, P squared, 2PQ. So you have to multiply by 215, 215, 215. Then you are going to get the answer for whatever is asked there, the expected genotypic frequencies. But the marks, I have to upgrade it, not to be 4. The question is clearly correct, but the marks is less. So in this case, when you are calculating the heterozygous, start by dividing, that is Q squared, find the square root, you'll find the recessive allele, -1, you'll get the dominant allele. Then, if you are asked about the expected genotypic frequencies, go to each value of Q, 0.598 squared times 215, you'll get expected genotypic frequencies for homozygous recessive. P squared, 0.402 times 0.402 times 215, you'll get expected genotypic frequencies for homozygous dominant. Then for the heterozygous, the same. 2 times 0.598 times 0.402 times 215, you'll get the expected genotypic frequencies for the heterozygous. But as far as we're concerned, 4 marks is just enough to find Q, to find P. It will earn you the 4 marks. Question number 2, it says, 9% of an African population is born with severe form of sickle cell anemia. They are homozygous recessive. Case where we saw an example of a selection model we are going to talk about. What percent of the population will be more resistant to malaria? Imagine if I'm setting the exam, I will not tell you because they are heterozygous. You are lucky there, I'm talking about heterozygous. I want to give you the number of heterozygous for sickle cell anemia, assuming it is in Hardy-Weinberg equilibrium, or all the assumptions for Hardy-Weinberg are kept intact. All the conditions are followed. So let's do again. 9%. They've talked about 9% homozygous recessive. And then they want you to calculate in the population how many will be heterozygous. 9 over 100. It means Q squared will be 0.09. This one is easy. Q will be 0.03, right? The square root, see, 0 will remain 0. Only 9. I want us to agree on the same note. Q will be 0.03. Just somebody to confirm so that we can move on the same page. The recessive allele, 0.03. Perfect. 0.3. Jenny? Ah. Oh. Good. 0.3. Jenny! Jenny, can you drink uji kidogo? For glucose ingie kwa brain. We jog around for another 30 minutes or so So that good, we are on the same page. 0 point. It is what? 0 point? It is 0.3. Oh, good. I can see somebody. Sorry. 0.3. Majority. I'm following the majority. Perfect. 0.3. Correct. Now, just not to go further, because we have P plus Q equals to 1. You will know P will be 0.7. But then you'll be asked how many will be heterozygous. 2 times 0.3, 0.7 times 100. So you'll have 2 times 0.21 times 100. 0.42 times 100. 42 of the population will be heterozygous. I think I got it right. When you multiply 0.3 times 0.6, we'll get 0.21. 0.42 times 100, because percentage, it has to equate to 100. So you'll have 42 of the individuals being heterozygous. So don't confuse. I don't need the dominant one. The people who can manage. I need that one who has a problem. The Q. We don't need the P. The P will be calculated after you've got the value of Q. Mostly if you start with the P, you are going to get confused and you're going to get wrong results. Some students will do Q and P at the same time. So sometimes it's good to be present during revision so that you don't get mixed up. Is it the testing again? Yes, it's still the testing, but then it will apply whatever we saw there. Question number 3 talks about ability to test a chemical. I think it just talks about the PTC. But in this case, we are talking about 500 students in the university, but 58 were found to be non-testers. So we are interested in the 58. Put 58 over 500. That is what I want. That is a Q squared. Divide 58 by 500. Then find Q. Do the square root. Find Q. After finding Q, subtract from 1. You'll get P. Then follow the question, whatever you've been asked. Calculate the percentage of heterozygous students, assuming the population is in Hardy-Weinberg. So from whatever you are going to get from heterozygous, multiply by 100, you'll know the percentage. Question number 4. A population of butterflies, color brown is dominant over color white. 81% of all butterflies are white. 81%. Any of these questions which will relate with heterozygous, please just do it like that. Nothing much. You have to get the square root because it's Q squared. 81 over 100. Q squared 0.81, which will bring us to what? 0.9. P will be 0.1. And then they're asking of calculate the percentage of butterflies, the population that are heterozygous, and frequency of homozygous dominant, allele frequency. Homozygous dominant 0.1. 2PQ, 2 times 0.9 times 1 will bring it towards. Then you will have 0.18 times 100, is it? But that's how we are doing it. So I'm just interested in the one which has a problem. Look at this, how it is twisted. And then even the next one, then I can leave. The PTC one, I can leave. Number 7, I can leave because it relates with the other ones. I think it's the same question. It says, number 5, in humans, the rhesus factor genetic information is inherited from our parents, but It is inherited independently of the ABO blood group type alleles, such that in humans, the rhesus plus or the rhesus positive individuals have the rhesus antigen on their blood cells, while the rhesus negative individuals do not have it. There are two different alleles for the rhesus factor, known as rhesus plus and rhesus. Assume that a dominant gene is RH, produces the RH plus. So it means the RH is dominant. And then it means the other one, which is RH without the plus, is the recessive. Assume the dominant gene is RH, which produces the RH plus phenotype, and the recessive is RH small ones, which produces the R minus phenotype. In a population that is in Hardy-Weinberg equilibrium, if 120 out of 500 are RH minus, so the story is just long, but the overall point there here is 120 over 500 is what you are going to work with. The Q squared there, the small RH, brings the RH minus, and is a recessive one. The story is long. Don't follow all the long story. Focus on whatever we want. So the Q squared is whatever is given there. And that is where the confusion was coming in. At a certain point when I said that exam to the students, they were struggling to give me the 380 out of 500. Nobody asked about the 380 out of 500. That one which has a problem, the fewer population which has a problem, that is what I want. So as you do the Q squared, do 120 over 500. Put it as RH minus, or the small RH. And talk about Q squared. You can put stroke Q squared, which goes to 120 over 500. Get the square root. You'll talk about Q. Then you'll find P, the RH plus one. And then you'll be asked what? The number of heterozygous. Because we are interested much on the traces of this recessive allele in heterozygous state. If it is low, then we are safe. We'll talk about the coming generations. Possibly this allele for the disease will go to extinct. It means it will have a fitness value of 0 in the coming generations. So we should not get worried. Good. Number 6. And then we'll go to the thalassemia one. I'll just explain. They are saying there are 100 students in a class. 96 did well. We are not interested in the people who passed. 96 did well, meaning 4 are the ones who failed. 96 did well in the course, whereas 4 blew it totally and received grade F until they say sorry. In the highly unlikely event that these traits are genetic rather than environmental, because it could be they did have enough books to study, maybe there was no electricity, maybe from their home where they come from, they joined during the day in class, but they have some challenges at home. Maybe the distance is long, but they are going to do the same exam. So they are talking about if the traits are genetic, the failing is genetic. Hata ukijaribu aje, it is not going to work. What if it was not environmental, those factors which I've explained, the difficulties they were going through in their homes, in their respective homes. But if it was genetic, they're saying, if these traits involve dominant and recessive alleles, and if the 4% represent the frequency of the homozygous recessive condition, calculate the frequency of recessive, they want Q, they want P, and the number of heterozygous individuals. It is simple. I need the 4 who failed. 4 out of 100. Do for me 0.04. Give me a Q of 0 point, is it 0.02? And then it means the P will be 0.98. Then if you do the heterozygous, 2PQ, 2 times 0.98 times 0.02, you'll be able to get the number of heterozygous. So in that case, we are talking about we need the Q squared. I don't need the P. The P are safe. Why should I mind about people who are not sick? They don't have a condition which is disturbing. I only need the ones who failed. That is the one we calculate. So don't start calculating the ones who did not fail. So even for thalassemia, they talk about a disease characterized by reduced absence of globin chain. They are explaining the disease. But the most important thing here is 1 in 500 individuals among Mediterranean have the disease, assuming it's in Hardy-Weinberg. Kindly take 1 over 500. That is Q squared. Find the square root. You'll find Q. Minus 1 from the value of Q, you'll get P. Continue with the questions. Frequencies of the two alleles. You already have P and Q. You have your marks. Percentage of heterozygous, whatever you get, multiply by 100. They talk about question number 9. A very large population randomly mating laboratory mice, because we have to maintain that. It is in Hardy-Weinberg. So, Kiprotich, we are discussing about genetics today with MBChB students, and we are almost. So if it can be of help, yes, it's still biochemistry, but genetics is not biomolecules. So we are talking about randomly mating because they have to maintain the Hardy-Weinberg equilibrium. Now, 40% white mice. White coloring is caused by a double recessive genotype. So it means Q squared is 40 over 100. Do 0.4. Q squared. Q will be 0.20. It means P will be 0.8. Calculate the allelic and genotypic frequencies. So if you do the allele frequencies you already get for P and the Q, the genotypic frequencies take P squared, Q squared, 2PQ, then you can put the frequencies there. That's how simple it is. So in the calculation of heterozygous, you have to start with the Q squared. Get the square root for you to get the recessive allele. Find the P. Go back to the question. You'll be able to solve it. Multiallelic, I'm almost finishing. For the ones who feel that they're really tired, I'm finishing. I'm going to do one question. We are going to do it together as you give me the answer of the calculations. Then it's going to apply to the other ones. Question number 11 and 12. So let's do 10 in the multiallelic Hardy-Weinberg. We are saying in a population, remember, we said in multiallelic system, we have P, Q, and R. P, Q, and R. You'll have P squared, you'll have PQ, you'll have PR, you'll have QP, you'll have Q squared, QR, RP, RQ, R squared, meaning the genotypic frequencies will be P squared, 2PQ, Q squared, 2QR, R squared, 2RQ. No, not 2RQ, 2PR. Correct, they have to appear 2. 1p, 2p, 1q, 2q, RR, correct. So this one should add to 1, the genotypic frequencies. Allele frequencies, P plus Q plus R should add to 1. So the question says, in a population of 800 people, 200 are A blood, 25 are blood group B. Calculate the alleles and the genotypic frequencies. So if you do A, it means P squared is 200 over 800. We want P. Do the square root. Then we have which one again? B is 25. B is Q squared, 25 over 800. Q equals to. Can we do that? I want us to divide P, 200 over 800. We find the square root. Somebody? We start with P, then you can just tell me P, 200 over 800. We can't see. Oh, sorry, dear. Sorry. We can do this one. Yeah, sorry. Can we divide? Somebody to divide Q squared? Correct, 0.25. Then we can find the square root of P. You know, this is P squared. Now we need P, Q squared, then Q. I'm waiting. Okay, 0.5. Our P is 0.5. Correct? Thank you. Can we divide 25 over 800? Divide first, then I can know Q squared. Yeah, thank you. That is for P. Can we go to Q? Thank you. Do for Q squared, divide first. Then you can do square root. Good. Jenny, is that the square root? Good. 0.11, 0.117, then Q 0.031, 0.031. Can we stop at 2? No, at 3. 0.013, correct. Now, we have to meet the rule. The alleles, 017, okay, 77, 01, thank you. Now, we want to meet the rule of P plus Q. We want to meet the rule of P plus Q. So you'll have P is 0.5. I want to wrap the Punnett square, but the genotypes will remain. So we have P is, I'll focus it here so that students can see. 0. Let me see if I write it well. 31. Yes, thank you. So we want our P is 0.5 plus 0.0313. That is P plus Q plus R equals to 1. So if we add, Can we add that one? We want R, which is 1 minus. Q is 0. No. Wait. Q squared is the 0.0. Q is 0.177. Okay. Okay. Sorry. So we shift. Let me see. Thank you for that. Because I rely on your answers. 0.013. So this one is 0.177. Meaning adding that one is easy. It will be 0.677. Correct? So R is, did I add it right? Okay. Okay. 0.46. No, it will exceed. Mohamud Hussein, if you say that it is that, first I want you to tell me if whatever I've added is correct, because from what I see, the total for Q and P will be 0.6. So R cannot be 0.4, which you are saying. Good. R has to be 0.323. Thank you. 0.323. Hussein, can you check? Please just check. We have to be on the same. Correct. So 0.323. Now, we want to find the genotypic frequencies, and we agreed. Thank you so much. 0.323. We agreed that the genotypic frequencies should add to 1. It cannot go beyond. Only at the point where we are doing the chi-squared of fitness test, where we think that there is a force, an evolutionary force, which has worked on our population. So let's go straight. You are going to multiply. We have 2PQ. We have 2QR. We have 2PR. Then we can finish with R squared, P squared, and Q squared. Of which Q squared you already know. You already give me 0.0013. So we are not going to look at for them again. 0.25. Remember, all this we wanted to add it to 1. They all have to add to 1. 0.0013. See, that was the Q squared. 0. Can somebody repeat for me the Q squared before we can look at the R squared? Q squared is 0.0. Is it 0.00 or it's 0.013? Kindly. Q squared was, there is no 2 zeros. Q squared 0. Yes, 10. Thank you. Yes, 0, 3, 1, 3. Because it has to add to 1. Whatever I've written there, if you can see, it's correct. Perfect. 0.0313, correct. Can we do somebody to do 2PQ up there, 2QR? You just tell us that is 2QR, 2PR. We already have P squared and Q squared. Can we now work on each of them? 2PQ, 2QR, 2PR, R squared. 2PQ, 0. I hope we'd multiply the 2. I want another person so that we can agree in unison. Somebody has found 2PQ as 0.177. which looks similar. Why does it look similar with Q? Correct. That is the max, because that is the answer. 0.177. 0.177. We can proceed. So we have to add a 0 after that because of our Q squared being 4 decimal place. Yes, agreed. 2PQ. 2QR, we have to add to 1 because we want genotypes for each. Yes, correct. That's agreed. 0.177. Can we move to 2QR? 2QR. 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 I'm waiting. Maybe somebody can do 2PR and then R squared. 2QR, 2PR, R squared, then we can add. It has to total to 1. Which one is that? I'm trying to look at. Yes, somebody has just told me now, 2QR. That's nice. Because the other ones are written and I don't know. 0.1143. 0.1143. 0.1143. We are doing well. 0.1143. 2PR. Somebody has already wrote the answer. 0.323. 0.323. R squared. R squared. R squared. Let me just confirm what I've written. 0.323. R squared. PR. PR. Yeah. 0.323. They've already written. I've already written. 0.104. 0.104. We can add now. Let me put these ones well. 0.250. 0.250. 0.0313. We are adding. They have to add to 1 so that you can know how correct they are. So let me add 0 at the end of this. This is 3. 11, 14, 18, 2. I want to see. 9, 10, 12, 17, 18. It is you people who gave me the answer. Is it that one? Is it 18? 1, 18, 1, 2, 3, 6, 7, 9. My friend, there is something wrong somewhere. What if we leave them to 2 decimal place? Let me see. 0.18, 0.11, 0.32, 0.10, 0.25. I'm leaving them at 2 decimal place. 0.31. Let me do that. I see. 8 plus 1, 9, 11, 16, 17. Carry 1. I don't know where the things are wrong. 9, 11, 16, 17. Carry 1. 1, 9, carry 1. What is 1? Wait, 17. 1 plus 1. 2, 3, 6, 7, 9, 12. It can't be like that. It has to be. What is that 0.99 land-owns? Maybe I can change it there. What is 0.99? The total comes to 0.99. Maybe I'm adding my own things. That is 1. So that's fine. But then you have to check. R squared is 0.10. Yeah, that one is 0.10. Okay. Good. Thank you. Hussein has also confirmed 0.99. Okay. No, that's fine. Because I knew when I was giving the examples, it has to enter to 1. 0.99 is 1, so it's okay. But maybe I'm the one who added well, in a bad way. Sorry, not well, but I confused some point. But now it is going to apply to all the multiallelic. Look at the 2 alleles which are represented there. They are squared. Do the square root, find individual alleles, then find the genotypes. That's how you are going to be asked the question. You have to find, because marks has to be scored for the 2PQ, the QR, PR, R squared, and that one you are also fighting with time. Calculations. And I said, don't ask anybody for calculator. We are not going to share calculator in the exam room, even in the cut room, kindly. If you don't have, then you can stay quiet until we finish the 2 hours. That's why I started early by saying, look for a calculator. We can't share calculators. That is where the papers are going to be passed. And I a calculator. I'm asking a calculator from so-and-so. If you don't have a calculator, I'm not going to allow you to share. Because we are going to calculate from the MCQs to the short answered. So why would you make life difficult for a neighbor who prepared well to have his own calculator or her own calculator? So let's have our own calculators. So it's going to apply well for question number 11. Number 12, do the same. Look for the square root. In this case, for number 11, we now have Q and R, no P. So we have to look for P. In question number 12, we now have P and Q, no R. So you have to look for R. So for the genotypic frequencies of the population, do you know? You are only going to multiply with the total ones when we are going to the chi-squared of fitness test. The chi-squared of fitness test, that is where you are going to multiply with the total number, because you need expected frequencies. In this case, you don't apply anything. That one will only help you when you divide so that you can know the frequency for each of the alleles. Wizard, please don't multiply anything. Where you use 800 or the total number of people in the population is when you want to get the individual alleles, because it's now in squared and we need to get the square root. Don't confuse them, please. And that's why we had to have this revision a bit early so that we can prepare well. Hardy-Weinberg assumptions, nothing much to talk about because it is notes which you can understand. But I'm going to stress on some points. Effect of small population size. Because we said that the condition is, the assumption of Hardy-Weinberg is, the population has to be large. So in the case of small population size, please don't talk about stories. Sciences do not want stories. Go straight and explain the two phenomena: bottleneck and founder effect. Simple as that. Explain for me the bottleneck and founder effect and give the example. You saw the founder effect, the Crevald syndrome, the one with the extra finger, the 6 fingers, how it came about, founder effect. Bottleneck, you saw about whatever happened in that island. People were swept. It could be a disease, any of those which can strike. Survivors were 30. But now the island, the allele for the disease, the percentage of the people who have it is many. Explain those two. Don't go deep into what genetic drift is all about. I need the two, bottleneck and founder effect. Simple as that. Short notes on selection models where both homozygous dominant and recessive are selected against. Heterozygous selected for. Heterozygous selectively advantaged. Homozygous dominant and homozygous recessive selectively disadvantaged. And the example there, you'll talk about the sickle cell anemia. The model, the homozygous dominant, they will die of malaria. The sicklers, homozygous recessive, will die with those symptoms related to the sickle cell, the bacterial infections, whatever, all those complaints, but they'll die of sickle cell anemia. The ones who will survive are the heterozygous. By giving the example of the disease and explaining much how the model works. That's what I want. Homozygous dominant selectively disadvantaged. That one is the example of Ngugi is asking something. So what's the difference between allelic? Oh my good Lord from heaven. Ngugi, I'm not going to answer that question. You have to go and read. I'm not going to answer. Only if you are not in class. I'm sorry. I might sound a bit weird, but I'm not going to answer that. I'm not going to answer. Maybe one of you will answer her. Somebody will answer Ngugi. I'm not going to answer that. Truly speaking, I'm not going to answer that. Ngugi, have you been in my class when I started teaching genetics until now? Then that question should not arise there. Now, I'm saying that if you talk about the selection model where, Eugene has tried to answer. So P will be the dominant one, the Q will for the allele, but then the genotypes. If he's still asking about the genotypic frequencies, she should know the genotypes and the alleles, how we started off with the genotypes and the alleles. So when you talk about that model, the question number 15, talk about genotypic. Don't do the genetic frequencies, genotypes. They are genotypes, correct? What Eugene is saying is that. So you'll be able to study more and know much about it. So the 15, the model gives much. The best example there which you can give me is the CCR5, where the homozygous do not have the traces for the CCR5 mutant allele. So in that case, they will die. The heterozygous have partial immunity because they have the traces of the recessive allele. But the homozygous recessive will survive. So you have to give me the model of the CCR5. We have several examples of the diseases which are supporting different selection models. So there are selection models and there are selection types. Stabilizing, directional, and disruptive. Those are selection types. But the selection models, we talked much about the genotypes being represented in the next generation, where we talked about the fitness value of 1, the ones which are represented in the next generation. For the inbreeding coefficient, we are going to do one example, and then you'll be able to apply in the second one. Question number 17. It's good to give example on the board, and then the students will be able to revise on their own as they follow the example which you've given. So we talk about inbreeding as one of the type of mating, of non-random mating. And we talked about inbreeding. The effect of inbreeding is, okay, inbreeding was mating among people who are genetically related. It could be due to cultural practices. It could also be due to small population size. That's why we talked about bottleneck and founder effect. At a certain point, based on the founder effect, these people move from a large population to a small one. Then they went to settle in an island. Now this population is small, but then people have to mate because they are human beings. So they'll have to mate amongst themselves. If somebody has a condition there, then as the population continue to grow, that allele for the disease will be spread to the coming generations. So at that point, we said that inbreeding will increase much of homozygosity. So we have to use the inbreeding coefficients. F was equals to 1 minus observed over expected. So if the inbreeding coefficient, I said if it is greater than 0.5, then that population is inbred. It means it's not in Hardy-Weinberg equilibrium. There is already one of those perturbations present. It could be there is non-random mating. So we are giving the example. We are talking about a data where we have the homozygous dominant 135 over, so A small a, small a, small a. You have to start by looking at the genotypic frequencies. That's what we said. So homozygous dominant, heterozygous, I don't know there how many, let me see. 135 plus 40, 175, 200. 200. Kindly, you'll give me the answer there. Students, you'll give me the answer. BB. What did I look at? AB, BB. It means this one is homozygous recessive, which we always refer to as, yes. So can we start with homozygous dominant? Those are the genotypic frequencies we are looking at. Homozygous dominant, heterozygous, homozygous recessive is the same as the MN blood group system, which we looked at. So can we do the 135 over 200, please? You give me the answer. We want to find if the population is inbred or not. 135. Divide by 200. Thank you. 0.675. Let us do 40 over 200. Thank you. Thank you, Hussein. Thank you, Charlie. Thank you, Mansa. And thank you, Sana. 0.2. Okay, Wangui, thank you. 0.2. We can do the last bit, 25 over 200. Yeah, thank you, Esther. London's, thank you, Natasha. That was for AB, heterozygous. Can we do for homozygous recessive? Homozygous recessive? 0.125. 0.125. And remember, they have to add to 1. So let's see and prove. Yeah, correct. Good, boys and girls. So we are going to find the allele frequencies. As you know, the P plus Q should add to 1. What we are going to do is obvious there. Half of the heterozygous. So it means our P is what? 0.775. Agreed? Agreed in unison? Yeah? Meaning our Q is, you just add 1 here, 0.225. Correct. That's agreed. We want to do expected heterozygous. Expected. Thank you. Expected. Expected. Hetero. Do 2PQ 2 times 0.775 times 0.225 times the total of number of people in the population, 200. Somebody to do that. Can you see? Yeah, do that, please. We have 2 times 0.775 times 0.225 times 200. We need the final answer there. Expected heterozygous. Then I come here, I do F equals 1 minus, observed is 40. Over expected, which we are going to look at. 1 minus, we are waiting for expected. Oh, good. 69.75, thank you. Still continue. Can you find it in decimal? Divide 40 over 69.75. Divide 40 over 69.75. Divide. Thank you, Michelle, Vinke, Hussein, Natasha. Thank you so much. Can we divide 40 by 69.75? We are almost there. I know it has taken 2 hours, but I know it will bring fruit. It will be fruitful for the ones who have been active from the start to end. 40 divided by 69.75. Perfect. 0.57. Let's do 0.57. F equals 1 minus 0.57. It means F equals to 0.43, meaning you do what? You have to say the population not inbred, right? That is less than 0.5. The population is not inbred. It means that population is heterozygous. So it follows Hardy-Weinberg equilibrium. If it is inbred, it means there is non-random mating. But from that, you can see we have a value of 0.43, which is less than 0.5. The population is not, put not in capital, not inbred. So you can exercise using that one or whichever you find in the book where you have the Hardy-Weinberg equations. You can try the inbreeding coefficient with that way, that formula. You have to find the genotypic frequencies first, which lead you to allele frequencies, because you need to calculate the expected. And the expected heterozygous 2PQ times the total number of people in the population. Then divide. After dividing, minus 1. Then you'll realize that it is inbred if it is more than 0, if F is more than 0.5. If it is less, then definitely that population is not inbred. This one talks about the chi-squared. When we talk about the chi-squared, it's more or less the same. It is more or less the same as what we have in the inbreeding. I'm going to use the first one. And remember, I told you, if the total number of the value which you get from adding all the 3 genotypes is more than 3.84, then reject the null hypothesis that the population is in Hardy-Weinberg equilibrium. So let's use the example 1, because the chi-squared if I might remember. It says observed minus expected squared over expected for all the genotypes. Observed. I think my phone is almost going off. It has stayed for long, so it's a bit dim. But I hope the students can see. I'm only remaining with a few minutes, then I'll be gone. So you have observed for each of the genotypes. Observed, expected, squared over expected. So what you have to do for question number 19, sorry, which will apply to all, you do homozygous dominant. We have 34 over a total of what? No, this one is 40, sorry. This is 40. Are they totaling to 75? If you look at that, you can see they're talking about cat friendliness. So they're trying to look at how the genotypes for several cats will be determined. So you will have the heterozygous 34 over 75. Then you will have homozygous recessive 1 over 75. Can we have the genotypes? Still, you have to start with the genotypic frequencies. Are we there? We can try one of these chi-squared, then I can leave you people to go and study. Or can we do the other one? Where it doesn't have the small ones, but still, I want us to do that one. Is somebody there? We can start with the homozygous dominant, 40 over 75. So we have to start with the genotypic frequencies. You get the allele frequencies. From the allele frequencies, calculate the expected genotypic frequencies for each of the three. Then you will subtract observed, whatever you can see there, minus expected over expected, which you've just calculated. Thank you so much. 0.533. I like the spirit. Let's proceed so that we can finish. Thank you. 0.533. Can we go straight to heterozygous? Thank you. Thank you all. Heterozygous, 34 over 75. 34 over 75. 0.4533. Yes. 0.453. The last one, 1 over 70. Yeah, it's like 0.00 something. 1 over 75. So they're asking if it is in Hardy-Weinberg equilibrium. That's how the chi-square test, fitness of test work. So we want to see if it's in equilibrium or not. 0.013. 0.013. Thank you all. So it's 999. Not bad. Not bad. Not bad. Maybe this one was 4, to be precise. So what happens here is, we are going for F, which is P. Can we get half of 0.53? 0.453, sorry. We get half. We want to add here. Q. Can we get half? Half of 0.53? Half of 0.53? Can we get half of that? Half of 0.53 kindly? 0.453. Half of it, so that we can add. 0.266. Agreed? 0.266. Somebody say 0.2665. I don't know whether we can do it 0.267. 0.267, which will bring what? 10, 9, 10, 8. So if you do 0.267, will it bring for us? If I do 0.267, with the upper one, half of this will give us 0.8, meaning we can do Q as 0.2. In other words, because we've followed half of this, you add to the full P squared, you'll get 0.28, sorry. So 0.2 is Q. So assuming P is 0.8, to be precise, Q 0.2. Now what you're going to do here is you are going straight to get expected genotypic frequencies. So you will get this one, 0.8 squared times 75. This one is this one. 0.2 squared times 75. Then this one, 2 times 0.8 times 0.2 times 75. Those are expected genotypic frequencies. Expected genotypic frequencies. After you've gotten the expected genotypic frequencies, substitute each of these. This is homozygous dominant. Again, for the heterozygous, do the same. Observed, expected, squared over expected, plus homozygous recessive, observed, minus expected, squared over expected. Simple as that. Again, the guy has said the half of that is 0.2265. Am I the one who wrote the wrong one? But now, that is what happened. So we have to get, Wizard is saying that the value which was given was different. 226,227. If I give 227, let me see. 227, 10, 5, 6, and his could be right because it has short so fast. So that Q, let us just add and work with whatever Wizard had said. 0.227, 1, 4, 2. Yeah, it makes sense. Correct. So we are going to correct it. Wizard, we have to correct it straight away. 0.76 squared, 0.2, 0.24. 24. Correct. So you see that one? Then this, sorry, 0.76. 0.24. Are we together? Wizard, that one, that is the correct one, which Wizard is saying. So I've corrected using the Wizard's number. 0.227. That's how it makes sense. Then the allele frequencies will balance. So after getting that, I'm just giving directives so that you can finish the remaining bit. The general formula for chi-squared fitness test is x squared observed minus expected squared over expected for homozygous dominant. Whatever you do, you have to do the expected genotypes down here for you to get the genotypic ones that are expected. Then you'll see, you will do now the value which you get for the homozygous dominant, but then you will start by doing a 40 minus the value for expected over the value for expected for the homozygous dominant. Repeat on how to get the expected genotypic kindly. That is what I'll put down here. You will not get expected before you get the genotypic frequencies. So you have to start with the genotypic frequencies, calculating whatever we've done there, what you are seeing on the board up there. After getting the genotypic frequencies, you have to get the allele frequencies for you to know how to get the expected genotypic frequencies. So allele frequencies, you will get full for p squared plus half in the heterozygous, the traces of p in heterozygous. And that's what we've done, and Wizard has just corrected us by finding the half, which is found in 2pq. After that, the expected there, you can see, the genotypes will always read as p squared, 2pq, and q squared. So p squared, you've already gotten the allele frequency for p being 0.76. Do 0.76 squared, whatever you see there, times the total number of people in the population, which is 75. Then do for q squared. Q, we found it to be 0.24. It has to be squared. If it is squared, then multiply by the total number of people in that population. Then you have to do the 2pq, 2 times p times q, times the total number of people in population. After getting the expected, substitute the observed at the raw data which you have for the genotypes. If the value is more than 3.84, because we are talking about a biallelic, reject the null hypothesis of population being in Hardy-Weinberg equilibrium. But you'll be able to check the genotypic frequencies. The values for the genotypes, they have to be in 1 to 2 to 1 ratio. So if they are more than that, you can talk of any of those evolutionary forces working on our population. So you will finish the remaining bits and you will apply to all which are seen in the chi-squared of fitness test. I'm sorry, I've gone 2.5 hours, but I believe it will pay back. So thank you for your time. As we prepare for the cut, kindly you read next Tuesday when I'm not in class. This Tuesday, not next Tuesday. This Tuesday when I'm not in class, you have to keep on revising. You'll find a venue the day for the cut. Vinke, you let me know. But we'll have the cut to start at 10. So thank you so much, and God bless, and all the best.